Hello, my name is Desiree L. Today we will be exploring the autosomal dominant disorder, achondroplasia. We will be discussing known facts of the disorder as well as discussing a human pedigree example that I have personally composed for this biology assignment at the University of Maryland Global Campus. Let's get started. For this slide, we will discuss what is known about achondroplasia. Achondroplasia is caused by a mutation in the FGFR3 gene on chromosome 4. It is a dominant disorder, so only one mutated copy is needed to cause the condition. Both males and females are equally affected, and the condition does not skip generations. The most common mutation, called G380R, makes the FGFR3 protein overactive, which slows bone growth, especially in the arms and the legs. According to the National Library of Medicine and Gene Reviews, people with one mutated gene show symptoms, while those with two normal genes are unaffected. Individuals with two mutated copies usually don't survive infancy due to several skeletal issues. The visual symptoms of achondroplasia, according to Mayo Clinic, include but are not limited to short height, disproportionately short limbs, large head, curved spine, and bowed legs. Other symptoms include spinal compression, breathing difficulties, and limited joint mobility. According to Cleveland Clinic, people with achondroplasia are usually diagnosed after birth by a physical exam and genetic testing of the FGFR3 gene. However, achondroplasia can be detected in utero by ultrasound. They are sometimes able to see the larger head with the disproportionately short limbs. There is no cure currently for achondroplasia. However, symptoms are able to be managed. Treatments such as limb lengthening surgery for some candidates, physical and occupational therapy, and monitoring threatening symptoms such as spinal compression and respiratory issues are available to people with achondroplasia. Although life with achondroplasia can be difficult, Mayo Clinic believes that with proper care, people with achondroplasia can live full and productive lives with a close to normal life expectancy. Here is the human pedigree example that I have personally composed for this presentation. As you can see, the following affected individuals are presented in generations 1, 3, as well as 5. In generation 1, affected male 1 marries unaffected female 2. Some of their children are affected and others are not, suggesting that generation 1, number 1, is heterozygous, and generation 1, number 2, is homozygous recessive. Each child had about a 50% chance of inheriting the disorder. In generation 3, affected female, number 2, marries unaffected male. Their child, number 2, is affected, while their other child, 3, is not, supporting the dominant inheritance pattern and generation 3, number 2's heterozygous genotype. Now, in generation 5, both affected individuals, 5 and 6, have a child, V2, who died shortly after birth. V2 was most likely homozygous dominant, which, according to NORD, is a lethal genotype for achondroplasia. Here are the references that I've used during the duration of my research and presentation of this project. Gene Reviews, Mayo Clinic, the National Library of Medicine, the National Organization for Rare Disorders, or NORD, and the Cleveland Clinic. These all helped me in completion and success of the research and human pedigree of the autosomal dominant disorder, achondroplasia.